I'm now going to introduce our next speaker, who is Soo Lee. He is a geochemist in the Chemical Sciences and Engineering Division at Argonne National Laboratory, and he specializes in synchrotron-based surface scattering. The title of his talk is Investigating the Structure and Reactivity of Mineral-Water Interfaces Using Synchrotron-Based High-Resolution X-ray Reflectivity. And you've got your talk all ready to go, so take it away.
Okay, thank you, Peter. Can you hear me?
I can very well.
Okay, thank you. Okay, hello, everyone. Thank you for the introduction, Peter, and the invitation to this workshop. And I'd like just briefly show you the overview, our research done by DOE-BES funding, and especially geoscience research program. And most of the work is done using the synchrotron source, such as advanced photon source in Argonne National Laboratory. So before I begin, I'd like to acknowledge the collaborators from the multiple institutes. So our work is done ourselves in collaboration with the University of Delaware, people in the University of Illinois, Chicago, and the Princeton universities. And all the research is done, most of them at least, at the advanced photon source, which is shown here, on the multiple beam lines, including sector 13, which is part of GSE cars. The topic or the research that I'd like to talk with you today is importance of ion adsorption on mineral water interface. Why this is important is because this process is controlling the mobility and the bioavailability of various elements such as contaminant and nutrient in the aqueous or natural systems. And then predicting or understanding this process, the impact of these materials or elements to the ecosystem is relying on the fundamental understanding of atomic scale process, how the ion adsorb or dissolve the interfaces of the mineral water in an atomic scale level. And this understanding is also important to understand other geochemical process in a low temperature environment, such as dissolution and the growth of secondary mineral phases at the mineral. So our approach to understand this process is based on simple cartoon shown in here. Let's say the mineral exposed in water, which contained ions. We can actually think about ion adsorbing on the surface with a certain rate, which is expressed by K adsorption. And some ion adsorbed might be dissolved from the surface, which is expressed by K desorption. And how ions will be adsorbed or dissolved from the surface is controlled by the balance between the dynamic balance between the adsorption process and desorption process. And the ratio of the adsorption to desorption will tell us about the affinity of ion. If the ion tends to adsorb more than the dissolve, there is a higher affinity or chance that ion will attract it to the surface. In reality, if you see the structure in an atomic scale level, the adsorbed ion distribution is much more complicated. For example, we can actually think about ion adsorbed directly to the surface as an inner sphere surface complex, which actually forming direct bonds to the mineral surface, whereas some ion might adsorb as an outer sphere complex, which is adsorbed species, but fully hydrated, which is similar to the species in the solution, but there is no direct bonding between adsorbed ion to the surface ligand. Therefore, it's normally assumed that this outer sphere complex is weakly adsorbed species, whereas the inner sphere complex is usually assumed as a strongly adsorbed species. So to understand these atomic scale features at the interface, we use the synchrotron-based surface scattering techniques. So thanks to Joanne's wonderful introduction. So I wouldn't go details about techniques, but X-ray diffraction is shown in here. So if you have a single crystal, which is shown in the schematic in the left side, we can measure X-ray diffraction as a function of angle of momentum transfer Q. Then what we observe in a three-dimensional reciprocal lattice space is a bunch of Bragg peaks. And the position of the Bragg peaks and the intensity will tell us about the atomic structure of the crystals. Now we say that we have mineral or the solid that has perfect surface, like mirror-like surface, then we can actually see the X-ray light is reflected from this mirror. We can actually measure how the intensity of reflected beam is recorded as a function of angle or one-term transfer Q. And the diagram on the right side is one example of X-ray reflectivity measured as a function of Q on the calcite water interface. And on the plot shown in the right side is basically showing the large variation of reflectivity, which is sensitive to the structure of the interface, so atomic distribution of water structure, etc. One thing that you can see here is the reflectivity itself is pretty low in terms of intensity. Sometimes it's down to the 10 to minus 10. So if you just plot the same data set into the linear scale, what we really care, the scattering from the intensity, scatter the intensity from the interface is basically in a background level intensity that we care. That's the reason why this measurement is so hard to do it in the lab source X-ray experiment and require a synchrotron, which has high brilliance, much higher brilliance than the typical X-ray source. Another benefit of using X-ray synchrotron X-ray source is that we can actually tune the X-ray absorption, X-ray energy to have element sensitivity or specificity. This is very similar to what Joanne described about X-ray absorption spectroscopy, but the difference is that we are measuring the signal, the spectroscopic signal, in a scattering geometry, and especially reflection geometry. Therefore, if you measure the data of the spectrum as near the specific, the X-ray absorption as of specific element, which we are interested in, if there is no element at the interface, then the spectrum should be flat because reflection of this spectrum is based upon how ions are distributed at the interface. But if there are some resonant atom at the interface, we can see the wiggle of the spectrum at the near the absorption edge. And the size of absorption edge will tell us about how much ions, the specific elements are at the interface. And the shape of a spectrum will tell us about where the resonant atom is located. So this is the direct proof of the distribution of ion at the interface with atomic scale level. So here I will show you the couple of examples of application of synchrotron X-ray reflectivity for the determination of interface structure between mineral water interface. Specifically, we're interested in the ion distribution on the charged mineral surfaces. So we measure high resolution X-ray reflectivity as a function of Q and analyze the data to get some kind of distribution or electron density distribution at the interface. And this specific feature is analyzed using non-resonant and resonant spectra that we measure, scattering data we measure, and we can probe the distribution of specific element at the interface. So the example that I show you here is mostly based on the muscovite mica, which is phyllosilicate mineral. It's abundant in nature, and it has very similar structure and chemical composition to many clay minerals, which are one of the most dominant species in soils and sediment. So the diagram or schematic representative of mica is shown here. And the top surface of the mica, which is basal surface of the mica, has negative charge on it. So that you can actually attract the cation on the solution by electrostatic attraction. So therefore, what we want to do here is by using X-ray reflectivity, we will try to look at how the edge of the ion are distributed with the sub-angstrom scale resolution. So one set of examples that I'm showing here is 4 different electron density profiles derived from X-ray reflectivity measurement done in water containing 4 different elements: rubidium, strontium, mercury, and zinc. Just briefly, this is electron density profile showing as a solid line, which is total electron density, including everything like water, edge of ion, etc., which is compared with dashed line, which is the electron density profile measured in DI water. So we see the difference in electron density indicating change of interface structure due to the adsorption of ion. The thing that I'd like you to concentrate on is this shaded area. This is the distribution of the specific elements such as rubidium, strontium, mercury, and zinc. What you found most critically is that the distribution of adsorbed atom depends on what types of element we have. For example, rubidium adsorbs mostly as a single species at around 2 angstrom from the microsurface, whereas strontium adsorbs as a 2 species in about 1.5 angstrom and 4.5 angstrom. Whereas zinc adsorbed farther as a single species, but much farther from the surface than the rubidium. Okay, there's difference in adsorption height and the speciation at the surface. Now I compare and determine what species is actually correspond based on the adsorption height. So we can actually think that these ions adsorbed very close to the surface as an inner sphere complex. Again, inner sphere complex is species that bind directly to the surface, whereas species farther from the surface is basically outer sphere complex, which is fully hydrated, and because of intervening water layers, it cannot really approach close to the surface. What we see here is also shown is the hydration energy of each element as a hydration enthalpy shown in here. So rubidium has the smallest value, smallest magnitude of hydration energy, means that this is the least strongly hydrated element, whereas zinc has the greatest magnitude of the hydration enthalpy, meaning that zinc is most strongly hydrated ions. So in the case of more strongly hydrated ions such as mercury or zinc, what we found is the dominance of the outer sphere, adsorption of the outer sphere complex over inner sphere complex. Also, what you can notice here is a small amount of extra adsorption of zinc and mercury farther from the surface. To make it clear, I make it 3 times bigger to show the distribution of ion, which should be the outer sphere complex, which is far from the surface because of hydration layer, but the distribution is much more extended from the surface than adsorbed inner sphere and outer sphere complex. Therefore, we call it extended outer sphere complex hereafter. So now I'm plotting the fractional coverage of 3 species, inner sphere, adsorbed outer sphere, and extended outer sphere complex as a function of hydration enthalpy. So the diagram on the left side, element in the left side is ion, is the least strongly hydrated, and the element in the right side is element that are most strongly hydrated. One simple trend that you can see here is the decreasing of inner sphere fraction as we increase the hydration enthalpy of the element, which is pretty much easy to understand because the absorption of inner sphere complex requires shedding the water molecules around the ion to have a direct contact on the mineral surface. So the most strongly hydrated element is the less likely the ion adsorb inner sphere complex because of strong hydrogen shell. The similar trend is observed between 2 outer sphere complex, especially extended outer sphere complex actually increase the coverage as we increase the hydration energy. So there is also control of hydration energy on the adsorbed ion speciation at the interface. To explain this trend, I use this cartoon so showing the ions in the surface and in the liquid and the surface of the muscovite, which is fully hydrated before the absorption of any ions. Okay, so if the ion here is strongly hydrated, it's really hard to shed the water molecules around it. In this case, the ion might adsorb as an extended outer sphere complex, basically far from the surface. If the ion is moderately strongly hydrated, then what you can see here, there's some kind of the removal of water molecules, but still maintain a primary hydration shell around the ion and adsorbed as an outer sphere complex. To form an inner sphere complex, what we need is basically some kind of destructive movement, basically shedding the water molecules around the ion as well as disrupting hydration layer around the water molecule, mineral surface too. So this diagram shows the delicate balance between electrostatic attraction, which bring the ion close surface, and hydration energy, which keep the ion away from the surface. That determine the complex ion, adsorbed ion speciation at the interface. So now I move on to think about how these complex ion speciation would affect adsorption phenomena, like how fast the adsorbed ion adsorbed or dissolved from the surface. To do it, We use the time-resolved RAXL, resonant anomalous X-ray reflectivity. So basically, we measure the energy-dependent change of X-ray reflectivity as a function of time while we change the solution composition. In this case, very simple exchange reaction between rubidium and sodium. So it's going from rubidium to sodium is rubidium desorption process or adsorption of sodium. And the bottom part is going sodium chloride to the rubidium chloride. So basically looking at the rubidium adsorption process for exchanging sodium. The result is shown in the right side, and the top panel shows the concentration of rubidium and sodium in blue and red curves respectively. So the left panel is rubidium desorption process, and the right column is rubidium adsorption process. And middle panel shows the change of rubidium coverage during desorption and adsorption process. And during desorption process, we found about like 90% or 95% of rubidium desorption occurs after like 30 to 35 seconds of the reaction, whereas rubidium adsorption is completed around 20 seconds. So definitely rubidium desorption is slower than rubidium adsorption. We also obtained the change of adsorbed speciation, in this case, inner sphere and outer sphere rubidium species. In a rubidium chloride solution, around like 90% of rubidium adsorbed as an inner sphere complex, only like 10% is outer sphere complex. But during desorption process, we found adsorbed outer sphere complex actually increase while inner sphere complex decrease. So there is a change of adsorbed ion speciation occurring in the desorption process. Whereas the adsorption process, we found the ratio of fractional coverage of inner sphere to outer sphere is kind of maintained as a constant, meaning that during adsorption process, we couldn't really see the difference in the speciation by our experimental time resolution, which is around 1 second. So based on this result, we found that there are two different processes, path dependency and the variation on ion speciation during adsorption and desorption. Rubidium adsorption is occurring almost directly to the equilibrium structure, whereas rubidium desorption is two-step process where the inner sphere rubidium transits to the outer sphere complex before it dissolves fully from the surface. So based on these studies shows that like classical ion adsorption phenomena in an idealized mineral surface, in this specific case, muscovite mica. But we can actually improve our understanding on a more complicated systems. For example, we can do the similar level of research in minerals which have chemical complexities, such as solid solution, and trying to see the impact of chemistry over the surface reactivity. What we can also do in the future is looking at non-idealized mineral surfaces, such as small crystals of clay minerals or manganese or iron oxide, and looking at those surface reactivity of those minerals in the future. Another example is looking at the complex solution chemistry and impact on the mineral-water interface reactivity. For example, you can look at the interfacial reactivity in extreme pHs, low pH or high pH, or the high salinity conditions in which ions are not really independent entries. They interact fairly rapidly and very readily because of increased concentration. Third example is looking at the mineral-water interface in non-ambient conditions, meaning that we can actually, for example, look at the mineral-water interface reactions in elevated temperature and pressure environment. This is important in particular looking at the CO2 storage strategy that we have, in which the injection of CO2 containing liquid involve the dissolution of primary minerals such as magnesium or iron-rich silicate phase, which ended up formation of magnesium on iron-rich carbonate solid phase, which is stabilized in the geological system. For this measurement, we actually need a combination of our expertise in X-ray reflectivity combined with some kind of high pressure, high temperature environment using, for example, diamond ambient cell as an example. I just want to briefly show what's the direction that we are going. one of the examples I showed is the impact of high salinity, in which the cation and anion interacts more readily than diluted condition. So again, this is electron density profile shown in the blue curve. And then the distribution of rubidium is shown in this shaded area from the RAXL. So I'm showing the electron density profile using some color coding. So the blue indicate the low concentration and red indicate high concentration. So we measure a series of X-ray reflectivity as a function of composition of solution, in this case, rubidium chloride, up to about 6 mole of rubidium chloride. What we see is a distinct change of electron density near the interface. We see the 3 different phases. From the RAXL, we found that this high electron density peak and this oscillatory feature is due to the adsorption of first, second, and tertiary rubidium species on the surface, which is more enhanced compared to the rubidium species and 3 millimole condition. What we also observe is occurrence of the additional peak at 4 angstrom from surface, which is from our molecular dynamic simulation, is the species, the anionic species, such as chloride adsorb on the surface. So if it actually shows the coverage of rubidium variation as a function of concentration of ion in the solution, in a diluted solution regime, we found that rubidium coverage follows typical classical Lamier isosome. But if you go higher concentration, the coverage of rubidium starts increasing again. And it's more than the theoretical limit of the Muscovite, indicating the adsorption of rubidium is causing overcharging. And our molecular dynamic simulation shows that this overcharging is related to positional correlation between cation, which is rubidium shown in the brown ball, and the anion, which is shown in green ball, such as chloride or iodide. So we can do the similar measurement in the rubidium iodide, in which we saw the rubidium iodide followed a similar trend, but the difference is in the rubidium iodide condition, we saw the more complicated interfacial structure extending farther from the surface. And molecular dynamic simulation shows that this extended distribution of rubidium iodide is due to the positional relationship between rubidium iodide, which is not only forming 2-dimensional surface, but also forming over the vertical distribution that actually forming some kind of nucleation and the growth of rubidium iodide crystal, even though solution was undersaturated with respect to this crystalline phase. Just quickly summarize it. So we use synchrotron X-ray surface scattering to probe the atomic scale details of structure and reactivity of geochemical interfaces. Our current work is focused on looking at the interface structure in an ideal condition, as well as dynamics. Some of the further studies has been done to understand nucleation and growth process. And in the future, we can improve our understanding of complex geochemical interface by using more realistic materials in extreme environment. Overall, I'd like to emphasize these research goals are achievable because of the unique capability of synchrotron sources, such as APS, which will be enhanced through the arrival of the upgrade. And our success ultimately rely on the combined application of expertise of surface scattering measurement and our understanding and the development of expanded setups and approaches that are specialized for this very community. With that, thank you for your attention.
Okay, thank you very much for that really fascinating talk, Soo. I learned a whole lot.
Peter.
And for people who joined a little bit late, we're asking questions through the chat. So we have a question from Athena Chen. Interesting work and great talk. How large is the area that the beam scans the surface in terms of microns, or is it larger than microns?
Right.
Does the beam scan through the mineral interface and average the data? So there are really 2 questions there.
Mm-hmm. So first question is the size of the beam. So typical size of the beam, you can think about in an APS, at least, is about 1 millimeter. So we actually looking at the structure, the average structure of like millimeter by millimeter species. Mm-hmm. But upon the development of new optics, and the focusing optics, and specifically, we can actually go down the smaller size. That's the reason why we think that we have a capability to look at the smaller particles with a similar resolution.
Have you achieved success with clay particles?
Clay particles are interesting. So so the definition of a clay, of course, is 2 micron size and smaller than 2 micron. But some of the clay minerals, such as vermiculite, actually can grow the large crystal. And one of the difficulty of a clay mineral is the chemical complexity and structural disorder. But if you have a small beam, it's possible that we can look at this type of measurement in the clay particles.
Okay, Soo, we're right at noon. Can you answer Carolyn Pierce's question super fast? In your opinion, to what extent is the example you presented for rubidium iodide versus chloride driven by solution structure versus interfacial structure?
So the quick answer is that rubidium iodide, rubidium chloride homogeneously recreate in the solution about 8 or 9 more. So the homogeneous process is very identical to each other. But the interfacial process is different in that rubidium iodide has perfect crystallography match with the muscovite crystal. Therefore, this epitaxial relationship actually drives the growth of rubidium iodide at the face, whereas rubidium chloride have about 10% difference in crystal structure. Therefore, the growth of epitaxial rubidium chloride is minimized. It's not really enhanced at the interface.
Okay, thanks very much. There are 2 more questions you'll see in the chat, and maybe you could just respond to those individually. Can you do that?
I'll do. Yes, thank you, Peter.
Thanks very much.